Hello, everyone. Thank you so much for joining us today. My name is Jason Peterson. I'm the Director of Data Analysis Solutions Product Management at Velsera, and I'll be facilitating today's webinar. Today's webinar is titled Comprehensive Clinical Molecular Results Reporting with Velsera's Clinical Genomics Workspace. I'd also like to introduce Dr. Shohini Sengupta, Senior Product Manager at Velsera, who will be our presenter today. Shohini completed her PhD in computational biology from Washington University in St. Louis and specialized in cancer genomics in 2018. Her research was centered around creating novel, supervised learning-based approaches to help prioritize driver mutations in cancer. She also worked with integrating multi-omics sequencing datasets at the DNA, RNA, and protein levels to help inform cancer therapy and uncover trends in cancer patient cohorts. At Velsera, Shohini's work has involved creating streamlined secondary analysis solutions for clinical NGS assays, as well as leading a team responsible for platform data ingress and novel integration solutions. Currently, she's focused on improving the experience for clinical evidence review and helping create a more streamlined process for comprehensive molecular clinical reporting, which she'll be discussing more today during the webinar. Thank you, Shohini.
Thanks, Jay. As Jay mentioned, my name is Shohini Sengupta, and today I'll be talking to you about comprehensive clinical molecular results reporting, particularly within Clinical Genomics Workspace from Velsera. So I wanted to first introduce Velsera. So some of you have probably formerly known us as Purion, and within Purion, we have our flagship product called Clinical Genomics Workspace, also known as CGW, which is an advanced genomics platform that enables clinical interpretation and reporting. But additionally, within Purion, we also provide other professional services, such as assay validation services, as well as lab enablement services. But now we have combined with two other companies, UGenTech, which specializes in lab workflow automation, as well as Seven Bridges, which specializes in omics analytics. And now we have merged and combined to form Velsera, which now we can even have even more powerful solutions to within the clinical genomics world, as well as I wanted to introduce our mission as Velsera is to really connect different types of omics data and clinical care to power precision medicine and eventually help drive better patient outcomes. So I wanted to talk about the key main goals of today's sessions and some takeaways that I want you guys to get. So number one, we're going to identify the core challenges of biomarker interpretation and reporting from next generation sequence testing. And number two, we will understand how the Velsera platform can present users with out-of-the-box solutions and transparently present underlying evidence with clinical meaning. And number three, we will learn about Velsera's adaptive learning algorithms and how we can use those algorithms to really streamline decision-making during the case review process to really cut down on repetitive decision-making from case to case. So I want to talk about the end-to-end workflow within a clinical genomics workflow, and I want to tell you how Velsera can support you in different steps or aspects of this workflow to really help you support and establish and scale your genomics program. So we start out with collecting a sample from a patient, either through a biopsy or some other method. You process that sample. You then do extraction, like DNA extraction, do library prep, utilizing an assay of your choice, and then you sequence your results. And that first part of the workflow is what we call the wet lab. And then the second part really involves, utilizing secondary analysis computational pipelines to process that sequencing output and make meaning out of it. So that is where you perform the variant calling, the data visualization, annotating your variants, and then finally, prioritizing and figuring out which of those variants are actually clinically relevant and interpreting those variants clinically. And then all of that will culminate into a final report that you can sign out. So the majority of today's talk is actually going to focus on that second part of the workflow I just described, which is what we call the dry lab component. And specifically, I'll be talking about Clinical Genomics Workspace, or CGW, which focuses on the clinical interpretation and reporting aspect of the dry lab component. And Clinical Genomics Workspace is powered by our in-house knowledge base, which is manually curated to contain clinical information that is associated with various biomarkers. So I do want to also point out, even though I will not be focusing on much of the wet lab part of this workflow, that Velsera has in-house expertise to also support the wet lab portion. We have things such as assay validation services, which can help your lab acquire samples, analyze data, as well as conduct analytical validations and document those validations. Additionally, we have professional services that help enable your lab. So if you need help in securing reimbursement, we can help you there. If you are a new lab that's looking to get off the ground running, we can help design your laboratory as well as help you select the assay that you would like to use, help you build quality management programs, as well as prepare you for regulatory compliance. So as part of that first part of the workflow that I was talking about, you have to select an assay that you would like to utilize, and these can be like targeted gene panels depending on the disease indication you are interested in. So as part of that, we help you choose from a comprehensive menu of ready-to-deploy analysis workflows that we have integrated within CGW. And these integrated workflows support a variety of next-generation sequencing assays. So we allow sequencing output from these assays to be brought into CGW through data ingress, and then to be able to do variant calling and secondary analysis all the way to reporting. So that is what is comprised within our integrated workflow. And a lot of these NGS assays are from a variety of vendors. They are vendor agnostic. We don't just support assays from one specific vendor, but rather a variety of them, such as, like you can see at the bottom, Illumina, Pillar Biosciences, and Thermo Fisher. There are additional vendors as well. And all of these automated workflows are thoroughly tested workflows with clearly designed specification that provide robust support. And in addition to providing you a seamlessly integrated workflow, we also help you streamline your implementation. So we give you the necessary framework and materials for you to rapidly implement and adopt an assay. And this is, reducing that turnover time from the moment that you receive your sequencing output all the way to signing out your first report. So here's the menu of all of the assay products that we support. As you can see, we support the Illumina and Thermo Fisher sequencing instruments. However, we do have flexibility. If you want to use a sequencer that is not Illumina or Thermo Fisher, we do allow the ability to ingest that sequencing output and integrate it within CGW so that you can report out on variants within your patient. So here you can see that we support a variety of disease indications. You can see that there's solid tumor assays, there's heme malignancy assays, there's germline, there's somatic cancer assays, and on the right side, you can see all of the different assays that we support that come from a variety of vendors. Just to point out a few, we have Illumina's TSO 500 family of assays, we have Pillar Bioscience assays, we have Archer assays, we have Oncomine assays from Thermo Fisher. And I also wanted to point out a newly added assay to our menu of assay products was the Illumina TSO 500 HRD assay. And this assay really allowed us to identify genetic alterations within the homologous recombination repair pathway, which can lead to, identifying genomic scarring and genomic instability within next generation sequencing data, which is really exciting. And this can help us identify appropriate candidates for PARP inhibitor therapy. So I wanted to talk about some of the major challenges that emerge when you're trying to interpret NGS panel results. And I would like to also talk about how CGW addresses these challenges. So one of the major challenges that comes out of NGS panel results is when you sequence a patient, you get a lot of variants that come out in your sequencing output data. And there's this need to prioritize and triage all of those variants that are coming out. So all of those variants that are coming out, not all of them are biologically relevant or really contribute to the initiation and progression of your patient's disease. And you have to narrow down from your universe of variants that are coming out, what is actually biologically relevant. But after you do that, not everything that's biologically relevant are a realistic drug target or can be clinically actionable. So there's this need to really narrow down all of your variants into what is biologically relevant, and then even more so, what is clinically actionable. And then number two, after you narrow down which variants are actually clinically actionable, each variant may have a breadth of clinical evidence that you have to sift through, and not everything may be relevant to your patient. Number three, there's a lot of automated reporting systems out there, but a lot of these, they will report out some results, but does not take any kind of user input or doesn't allow you to make any user modifications and user decisions to be saved for the future. And because of that, this causes there to be repetitive work that is needed for when you see the same variants across different patients in the same disease type. Another challenge is a combination of biomarkers may have a different clinical meaning than its individual counterparts. It's possible when you see several biomarkers together in conjunction in a patient that they may have a different response to a drug than if you were to see those individual variants by themselves in the patient. Lastly, now that you've looked through and prioritized your variants and sifted through all of the evidence, there's this need to visualize all of that information, all of the relevant biomarkers together in one concise report. So now that I've talked about the challenges in NGS results, I want to talk to you how our tool, Clinical Genomics Workspace, can help you address some of those challenges. So the first way we do this is we help you prioritize and triage all of those variants to really hone it down to what is clinically relevant. So to do that, we provide automated classification of next generation sequence findings. So we within CGW implement the CAP-AMP-ASCO classification scheme. So for those of you who don't know what that is, this was a working group that was created with experts from each of those three societies, AMP, ASCO, and CAP, and they specialize in, clinical oncology and genomics, and they came up with a standardized way to classify somatic variants in cancer so that you could interpret them and report out on them. So in this classification scheme, there are different tiers. So you have tier 1A, 1B, 2C, tier 3, and 4. And within CGW, we calculate this classification for each variant. And on the right side is a depiction of the decision tree that CGW follows to classify a variant into one of the tiers. And we essentially use different pieces of evidence. For example, we consider for that variant if there's any drug label or practice guidelines available. And if so, we'd consider, is that guideline within the patient's disease or is it for other tumor types outside of the patient's disease? We also consider that for clinical trials, whether it's in the patient's disease or outside of the patient's disease. And additionally, we consider whether that variant is a common variant found in normal human population. And taking all of that into account, we assign the variant one of the CAP-AMP-ASCO tiers. Another classification scheme that we implement within CGW is called the ComproMed biological classification. And this is a classification scheme that's popular in Europe. And this was created by a panel of Belgian experts in cancer diagnostics. And this classification scheme differs from CAP-AMP-ASCO in terms of this classification scheme really talks to the biological contribution of the variant itself, and not whether there's any clinical information associated with that variant. This is talking about the pathogenicity of the variant in the disease context. And also this classification scheme, unlike CAP-AMP-ASCO, is independent of tumor type. So it's really just talking about the biological contribution of the variant. And here is a view of what our biological classification scheme looks like within CGW. And as you can see at the top, for the variant, you can visualize the CAP-AMP-ASCO tier that was automatically calculated for this variant. And then you can also see the biological classification, which is variant of unknown significance, VUS. And then below it, you can see all of the discrete pieces of evidence that feed into and determine that final biological classification. And the cool thing about this is that you can utilize both of these classification schemes and take the consensus or look at the two results to really prioritize your variants for reporting. And additionally, CGW has the flexibility to incorporate additional classification schemes in the future. So if there is a classification scheme that a lot of laboratories are using, we can also incorporate that in an automated fashion within CGW. So I talked about automated variant classification within CGW. Now I want to talk about how we help you prioritize variants after the automated variant classification. So this is what our landing page looks like in CGW. So if you run a case, after you run that case, you will see this all findings page that describes all of the variants that are in your case. And we do provide an automatic prioritization of these variants in terms of the order that it's displayed. As you can see, we prioritize obviously like non-variant biomarkers like microsatellite instability, tumor mutational burden, but we also order these variants based on their tier. So 1A variants will have priority over 1B over 2C. But we also give you the flexibility as a user to be able to reorder those variants so that you can set the order that you want to see these variants in your report. And I will demonstrate to you what this actually looks like in CGW. So as I mentioned, this is after you run a case, you look at your all findings, and this is all the variants that are in your case. And as you can see here, it's already automatically prioritized 1A, 1B, 2C. But if you do not like this default order, you can drag and drop and reorder these variants. And this will dictate the order of how these variants will present in your report. And I do also want to point out that there are filters on this all findings page that can help you prioritize or focus on certain variants. You can filter it by CAPAMP ASCO and then apply. You can reset the filters. You can search for a specific keyword, like if you're looking for a specific gene, like BRAF. So this is your all findings page. So in addition to helping you prioritize your variants, we also provide you clinical evidence right at your fingertips. So when you click on a variant on your all findings page, this will navigate to another page where you'll have a bunch of tabs like clinical relevance, clinical trials, interpretation. And you can go into your clinical relevance tab and visualize all of the clinical information that is associated with your variant. And we call these clinical assertions. And this information will have, the source that information is coming from. Like if it's a guideline, like from FDA or NCCN, you will see the disease that's associated with that clinical assertion. You will see the drug name and you will see the impact, whether it is responsive or non-responsive. And then we smartly, as our system default configuration, we do rank these assertions based on what we think you would like to see at the top versus at the bottom. So more important assertions that are relevant to your patient's disease, as well as from higher level sources, will be present at the top by default. And we also give you the ability to turn off any of these clinical assertions that may be irrelevant to your patient from the report, if you don't want to include all of these assertions on your report. So I will show you what this looks like within CGW. So let's say this is your landing page and you want to go look at BRAF P600E. So you click on that and you'll see all of these tabs, and we're going to focus on the clinical relevance tab. And this patient was for colorectal cancer. So now you can see all of the clinical assertions associated with this variant, and they're grouped by therapeutic significance, which has drug information and its corresponding impact. And then you can also see prognostic information, which will tell you if it's unfavorable or favorable. And you also have diagnostic significance assertions listed here if those are available. So you can click on any assertion, and this will pop up details on the right side that explains a little bit more about that assertion. And you can also see that these assertions were ranked in a default way, and this is what our CGW has configured. And here you can see like things that are within the patient's disease, colorectal cancer, those assertions are prioritized at the top. Higher level sources are prioritized. PubMed is lower, and things like FDA are at the top. We also try to prioritize things that have a responsive impact. So we do an automated way of ranking these to help ease your review of all of these assertions. Another thing I want to point out is we allow filtering, so you can filter by assertions that are related to your patient's disease, or you can filter by assertions that are in other tumor types. And then here, this is that toggle that I was talking about, where you have the choice to toggle anything off your report if you deem it not relevant to your patient. So you can just toggle them off. So I talked a lot about how we have automated configurations to help ease your review process, but we also give you the flexibility to configure your preferences as well. So if there's something in our default configuration that you want to change or modify, we give you the flexibility to do that. And I will talk about the different ways that you can configure your preferences. So how I just showed on that clinical relevance page that our system, our CGW prioritizes and ranks certain assertions in a certain way. You can also control how you want to rank those assertions and what factors are the most important in terms of ranking those assertions. Like if you want to determine that source is more important, or tier of that assertion is more important, or disease relationship is more important, you can define that and pre-configure those configurations prior to running a case, so that every single time you run a case, your preferences will be outputted in terms of ranking your assertion. Additionally, if you see on this screenshot on the bottom right, you see that there are some assertions that are turned off by default. And you can pre-configure if there are certain assertions, like certain sources, that you do not want to be in your report by default every single time you run a case. So let's say that you are a US-based laboratory and you're only interested in guidelines from US sources. Then you can specify European sources like ESMO or EMA to be turned off by default. And that's what you're seeing here. Additionally, we do display clinical trial information for your variant, and there's a number of configurations that you can do for clinical trials as well, similar to the clinical assertions. So now I will show you what clinical trials looks like within CGW. So let's go into a different variant. So let's go into this fusion, and you have your clinical relevance tab, and then here's your clinical trials tab. And when you click on that, you'll see all of the matching clinical trials that are within your patient's disease. And here you'll see the trial name, you'll see the disease, you'll see the phase, you will see the closest distance of the distance. So this is when you accession your patient case, you can specify a zip code of your patient or the zip code of your laboratory or medical center, and CGW will calculate what that distance from that zip code you accessioned is to the trial site. And that's what you're seeing here. And then you'll see the trial ID, and you can click on this trial ID to expose the actual clinical trial website. And then you can click on the icon to get additional details. So as you can see here, there's a ranking that's already in place. So CGW does do an automated ranking. But the things that you can configure is how you would like to rank your clinical trials. Like for instance, you can put more weight to if it's a higher phase or if it's a closest distance. And these are things you can configure ahead of time to drive how your clinical trials are ranked. Additionally, you can configure how many clinical trials you want to be in the report by default. So right now, this configuration has 7. So anything that's not in the top 7 will just automatically be out of your report. You can also configure like what source the clinical trials come from that you want to be in the report by default. So let's say you're, once again, a US-based laboratory, then you say that you're only interested in US-based clinical trials from CT Gov. So in this case, you will only see CT-Gov trials in your report by default, and anything from a different source like EU-CTR, which is European Clinical Trials, will be out of your report by default. So this slide, it just gives you a little bit more information in how you can configure those assertions on the clinical relevance page, and how you can feed it information to rank your assertions. And these are the 5 major factors that are taken into account in terms of ranking your assertion. And you can indicate to CGW which of these is most important to you and how you want to rank them. So the different types of information here is like classification, your drug impact, your disease relevance, whether your assertion is within your patient's disease or in other solid tumor types, the disease relationship of the assertion with the disease of the patient, whether it's an exact match, an ancestor, etc. And you can also determine what sources are most important to you, because in our automated prioritization, we value and rank FDA source higher than PubMed, but you can also switch around what you value and what you prioritize. So I talked about how you can configure your preferences to help you reduce on some of the repetitive work every single time you run a case. But I also talked about one of the challenges of a lot of times you see the same variant present in multiple patients in the same disease context. And you might have to do some repetitive work or user-reported decisions every single time you see that same variant. But within CGW, we can remember your decisions so that you don't have to when you see the same variant in the same disease context again in a future case. And I will talk to you about some of the user decisions that can be saved and propagated for the future. So for instance, I talked about you can turn off certain assertions for your variant if you deem those to be irrelevant to your patient. So here is an example of like when you're turning off, maybe you want to turn off and toggle off any out of disease assertions because there's plenty of in disease assertions available. So you can save this and propagate for the future. So the next time you see that variant, these assertions will be turned off by default. You can also change the order of the assertions. So there is like a box with 3 dots which allows you to drag and drop and reorder these assertions. And you can also save that and preserve that order for a future case if you see that variant again in the same disease. You can write an interpretation for each of your variants, and that can also be saved for the future. Additionally, I mentioned how we automatically calculate a CampAmp ASCO tier for your variant, but it's possible that you might want to modify the tier that we calculate, maybe because you saw an assertion that you decided to turn off because you thought it wasn't relevant to your patient, and that might require you to update or down tier your CapAmp ASCO tier that was automatically calculated. And that tier modification can also be saved the next time you see that variant in the same disease context. So another thing that we provide that helps you remember your decision as well as helps you focus your review. So let's say that you've ran several cases and then you run a new case that has some of the same variants that you've already signed out and reviewed previously. So we provide variant level alerts on that all findings page that I showed you to help really direct your attention to all of the findings that need review. So for instance, these are the 3 different variant level alerts that you might see. The first one being an update tag. And this indicates that you have reviewed this variant before and signed it out. However, there could be new or changed evidence in the knowledge base that could affect this variant. And maybe you might want to look into this variant a little bit more to see what has changed. The novel tag indicates that you've never seen this variant before in this disease context, and this is the first time you're seeing it. So you probably want to spend a lot more time reviewing the evidence that is associated with this variant. And then third, you could have a familiar tag, which means that you've seen this variant before in the same disease context, but there's been no change, no update in knowledge base content, and that use everything that you've reviewed is probably going to be the same, and that you might want to spend less time on it because you've already signed out a case with the same variant. All right, so in addition to variant level alerts, when you click on a variant and go to that clinical relevance tab, there will be assertion level alerts next to the source, as you can see in the screenshot, to indicate what exactly changed since the last time you saw that variant. So let's say you, on a variant, you had an update tag, so you click on that variant, and these alerts will tell you exactly, which assertion was affected and what you need to draw your attention to. So for example, if you see a blue star, this means that this assertion is novel and it's been added to the knowledge base since the last time you reviewed this variant. The yellow icon indicates that this assertion has changed in some way since the last time you reviewed this variant in this disease context. And an example of what could have changed is, like, for example, there could have been an added source, or there could have been an added disease. So now I want to show you all of this in practice and what this looks like within CGW. So I'll show you several cases and show you how your user decisions can be saved and propagated for future cases. So in this first case, let's say that this is your first time running a case within CGW, and it's within non-small cell lung cancer. And you make some decisions on your variants and you sign out a report. So let's look at this first case. As you can see, the variant level alerts that I was talking about, everything says novel because this is the first time you're running a case. You've never seen these variants before. You never signed out any evidence. So you probably need to look and review at every single one of these variants. So let's look at BRAF V600E. So you go into the clinical relevance and you see that there's a lot of assertions reported within non-small cell lung cancer. But you decide that maybe you're only interested in the assertions that are related to your patient's disease, which is non-small cell lung cancer. And anything outside of that is not important. So let's turn off all of these assertions that are out of disease. And as you can see, as I turn these off, there's like this blue bookmark that appears. If you hover over it, it said this decision will be saved for the future. So if you have this blue bookmark selected, this indicates that you want to propagate this decision of turning off the assertion to future cases when you see this variant in the same disease context. So you can turn off all. And then now let's look at all of the remaining assertions. You see some that are non-responsive. And maybe you say, you want to turn these assertions off. But let me just show you the functionality of, like, maybe let's deselect this bookmark. So this indicates that you don't want to report these assertions for the current patient, but you don't want to propagate this decision for the future. All right, so you go back to your all findings page, and then let's say you're hypothetically done working on V600E. So a way that you can indicate that you're done reviewing it is you can check this box, which indicates to yourself and other users that are using or working on this case that you've reviewed this variant. So now you can move on to another variant. So let's look at this TP53 variant. So you go into TP53, you look at all the clinical relevance, and you see this prognostic assertion that has a 1B classification, but maybe you decide that this isn't relevant or I don't want to report this. So you turn that off. But because you turned off this assertion, which is 1B, this overall CAPAMP ASCO tier needs to be updated. It needs to be down tiered, and you down tier it to 2C. So you can confirm. And then another thing that I want to show you is if you go into the interpretation tab, so we have system generated interpretations here that help you like speed up that process of writing an interpretation. And we provide things like gene summary, prevalence summary, which has information from Cosmic. And then there's a concise shortened text that appears that describes each of the assertions that you have turned on in your clinical relevance tab. And this dynamically updates as you turn on or off assertions. And I can show you how that looks. So if you were to turn this back on, you'll see another sentence appears that's associated with that assertion. And this, the right side is system generated. You can copy this over to the left side, and then you can modify and edit the interpretation. So let's say that you modified this interpretation and edited it for this case. And then this will get saved. All right, so now, okay, so let's say you sign out that first case and now you run a second case in non-small cell lung cancer. But now let's say that in between when you ran this case and the first one, there is newer content available in your knowledge base. And you are running the same exact variants that you did in case one, and it's the same exact disease. So now I want to show you how the user changes that you did in the first case are preserved in the second case. So as you can see, now there's these variant level alerts. They don't say novel anymore, but you see a lot of the variants say familiar, which means you've reviewed and signed this out, and then the knowledge base content that got updated does not affect these variants. So you probably don't need to focus on reviewing these variants again. However, this BRAF V600E variant, you see the update tag, which means the updated knowledge base content that occurred might have affected this variant, and you might need to re-review some of the assertions within it. So if you click on that variant and you scroll down, you can see that there's an alert on this assertion. So this indicates that this assertion was changed. And if you click on it, you can see exactly what was changed. So there were a couple of disease terms that were added for this assertion into the knowledge base. And as you can see, if you were to filter by out of disease assertions, all of these are already turned off by default because you had turned these off that first time you saw it, and it's in the same disease context, and these got propagated for the future. The only thing that's not turned off was this assertion, which is turned on because this assertion was affected and updated because of the new knowledge base content. And because of that, it's telling you you need to re-review this assertion and decide whether you actually want to turn it off or turn it on for your current patient. And then if you go to your patient disease assertions, remember we turned off the 3 PubMed assertions because they're non-responsive, but we actually didn't select that blue bookmark, which indicated I don't want to save this for the future. And as you can see, this decision was not retained or saved because you only wanted to save that for the current case and not future cases. Now we're going to look at that TP53 variant. And as you can see, you had changed this from a 1B to a 2C, and that tier modification has been preserved, as well as the assertion that you turned off, that has been preserved as well. And then if you go to interpretation, you will see the edits that you made to the interpretation are also preserved. So on your left side, if you made any user changes for a variant in the same disease, then you will see that on the left side when you see that variant again, and those interpretations are preserved. All right, so the third case I want to look at is a case in soft tissue sarcoma. So let's say that you ran several cases in soft tissue sarcoma previously before this case, and you signed it out, and some of the same variants are also in this third case. And the only difference is there's some newer content that was available in between when you ran this case and the previous cases. So let's look at what that looks like. So as you can see, 2 of these variants were affected by the knowledge base content update. 1 of these were not. So let's look at this fusion transcript. So when you click on here, you see 3 assertions were affected by the new knowledge base content. And when you click on this, you can see there's a blue icon, and then you see this is grayed out, and then there's this yellow icon. And if you look at these 2 assertions, they're essentially the same disease name and the same drug. The only difference is the source. So what happened here is this was a PubMed assertion, which NCCN decided to adopt that and put it into its guideline. And because PubMed is a lower level evidence, this was deleted and removed by our curators from our knowledge base. And that's why you see this grayed out, because this was removed since there's higher level evidence now available for the same drug. And then if you click on this, you see the yellow icon, and you can see that there was a disease that was added to the assertion that caused this to be different. Okay. So one of the challenges I had mentioned earlier was being able to report on a combination of biomarkers. So within CGW, we do give you that capability to visualize and interact with evidence for co-occurring variants in one place, rather than having individual biomarkers all with the co-occurring evidence attached to each of those individual biomarkers in multiple places. And as you can see here, this is a screenshot of a co-occurring variant biomarker. So we have a MET copy number gain and a RET fusion. And combined, this may have a clinical evidence attached to this combination. And as you can see, this is the combined co-occurring variant, and then below it is just the fusion by itself. And as you can see, both of these have the same drug associated with these biomarkers, but they have different drug responses. And the drug response is non-responsive for the co-occurring pair, and it's responsive for just the fusion itself. So this is a prime example of where you could have differing response depending on whether you have a co-occurring variant with the fusion. So I will show you what this looks like in CGW. So as you can see, we visualize the individual biomarkers separately, and they have their own CAPAMP ASCO classification scheme. But then you see another entry for the co-occurring biomarker, which could have a different biological relevance or a different drug response for different clinical assertions. So if you click on that, you can visualize the assertions associated with this combined biomarker. And if you look at the individual biomarkers, they have different associations clinically. And that's the fusion, and then this is the copy number, which also has a different set of clinical assertions than the co-occurring biomarker. So I also wanted to talk about our clinical sharing network. So we have a vast network of a lot of laboratories that utilize CGW across the globe. There's about 140 sites, as you can see on this map, that utilize CGW, and they sign out hundreds and thousands of interpretations. And that really creates a rich resource of data and interpretations that we actually have incorporated within our knowledge base. And we expose that data to current users, future users. So when a user goes in, you can see all of those shared interpretations that other people have written. And a lot of our users utilize this shared interpretation data as kind of a kickstart to start writing their interpretations for variants that they've never seen before, or as like a gut check to make sure that the conclusions that they're coming up with match what their peers have written about a certain variant. Additionally, this rich source of data can provide you information about how often you are seeing a variant in your lab versus other laboratories. So a lot of my presentation talked and focused on variant biomarkers, but I also want to make a note that we can support reporting non-variant biomarkers from next generation sequencing, such as like TMB values, like tumor mutational burden, or MSI, microsatellite instability, we can visualize those values and put them on a report. But additionally, our knowledge base has curated content for TMB and MSI so that you can see clinical assertions that are associated with different TMB and MSI values. We also support reporting non-variant biomarkers from other molecular tests besides next generation sequencing. You can report things such as like FISH and IHC, as well as upload a picture for those tests and put them on the report. So our report's very flexible in terms of incorporating results like non-variant biomarker results. So previously I had mentioned, there's this need to be able to concisely visualize all of the important biomarkers in your case and put them into one concise report. So this is a screenshot of what our report looks like in Velsera. And as you can see, this first page has a summary of all the important findings within your case. And then the following pages of this report kind of expand on, the summary and give like your interpretations that you've written out per variant or like the assertions that you've had it. It's a more detailed look. So now I want to show you what that process of report finalization looks like within CGW and how you can visualize your PDF for the case that you've done evidence review for. So now let's say that you finished reviewing all of your variants and now you want to sign out your report. So you would click on preview and finalize. And when you click on that, you land in your report finalization page. And you can see these different text sections. There's executive summary, pertinent negative, and then just a third placeholder section. But all of these text sections you can pre-configure. So if there are certain sections that you're interested in showing up by default every single time you run a case, you can configure that ahead of time. So like, for example, if you want to have methodology or disclaimer or any kind of test details, you can configure those names as well as the default text that you would like to include. And that will show up in this report finalization page. And you can go in and edit the text so that it corresponds to the current case that you're working on. So now you can also visualize what your report looks like right next to your report finalization text sections. So if you wanted to edit or create your executive summary based on what's actually in your report, you can visualize that on the right side and write it up at the same time. And anything that you change on the left side will dynamically update on the right side when you refresh. So here, as you can see, the report has like your patient details, your report summary, which is the executive summary that you had put in here, and then a summary of all of your important findings. You see a clinical implications section that has all of the different assertions that you had turned on in that clinical relevance page, and also a summary of the number of clinical trials. Your next section includes interpretations that you have written out for each of your variants. And then you have all of the clinical trials that you had turned on for each of your variants, and you can see what variant they're associated with. And then your TMB and MSI data is also present in here. So these are the non-variant biomarkers. You can see their values, their quantitative, their qualitative value, their clinical impact, the assertions, and also the interpretations that you've written for each of those. So a lot of my presentation talked about the clinical genomics workspace and how we provide you this seamless end-to-end workflow and help you prioritize your variants and be able to create a report that you can customize. But I do want to reiterate, and this is something I'd mentioned previously, is that we provide a number of professional services outside of CGW that can help you. And some of these things are, we can help you facilitate a successful lab launch if you are a new lab, as I mentioned, helping you set up your lab, building a quality management program, selecting your assay, etc. We also provide those assay validation services that I had mentioned previously, where we help you with experimental design, analysis, and documentation of your validation. And then lastly, we also help you with revenue cycle management services by developing and helping you with a reimbursement strategy based on your region and your assay. And lastly, we have clinically trained experts that can help you with your case review process and help you write interpretations and sign out. And with that, that is all of my slides. I would like to thank all of you for attending today. And let me know if you have any questions. There's also, if you have a question, there's a Q&A box if you want to put your question there.